Hello, Senior 2, how are you? Today we are going to talk about the heterotrophic nutrition. Heterotrophic nutrition is the other type of the nutrition. The first one was the autotrophic nutrition, where organisms can make their own food. What the heterotrophic nutrition, these are organisms, this type of nutrition, organisms do them where they depend on other organisms to feed. Okay, the heterotrophic nutrition is another type of nutrition where they depend on another organism to feed, which already fed on plant or they feed on plant directly. Okay, in the heterotrophic nutrition, we are going to talk about the digestion, where the digestion they need system where they can feed and take the benefit of what they eat. Okay, okay, where the digestion is the conversion of large food substance like the polymers into smaller ones like the monomers by hydrolysis. Hydrolysis means by the help of water. This process cannot take place except in the presence of enzymes, okay, where large and complex substances such as starch, proteins, fats, with the help of the enzymes, where they are catalyst, they work as a catalyst, they catalyze the reaction, help to fasten the reaction, converts these large molecules into simple and smaller ones, where the starch will be converted into monosaccharide, the protein will be converted into amino acid, the fats will be converted into fatty acid and cholesterol. Okay, this, in this we are going to talk about the enzymes. What are the enzymes? The enzymes, they are protein substance. They are protein in nature. Okay, they are formed of protein, where the protein is formed of amino acids holding together with the peptide chain. Okay, these enzymes where these enzymes are protein substance have the properties of a catalyst. يعني they can help enzymes catalyst a helper to the reaction, as they can accelerate the reaction. يقدروا increase the speed of the reaction without taking part in the reaction, without participating in the reaction, only as a transporter. بس this transporter make the reaction faster. Okay, and these enzymes also they are so specific, have the ability to activate particular chemical reaction, only works for substrates, special substrates, to get the product without any change in the enzyme structure. Okay, where the enzyme looks like that, a key and its lock, a key and its lock. This is the enzyme, and this is the active side of the enzyme. The active side in which the substrate or the products or the reactants will work, will combine with the active side of the enzyme to form a intermediate compound called a enzyme substrate complex, where this intermediate compound will be then broken down into enzyme and the product. It will convert the reactant into product. Okay, so this is the action of the enzyme. Okay, the enzyme again, where the reactants again combine on the active sides of the enzyme. Okay, this will help to the formation of a protein without the change of the enzyme. Look here, الانزيم ايه زي ما دخل زي ما طلع, has no change. Okay, this is how it works. The enzyme or the active site and the substrate, they combine together, and then the enzyme works on the substrate or the reactant and form the product. Then it will remove the enzyme will go to make another job, the same job again, and the products are produced. Okay, These are the enzymes. How can these, the mechanism of working of the enzyme? Then what are the properties of the enzyme? As I've said before, the enzymes are protein in nature. They are protein. And you have to put the last year that the protein is formed of amino acid groups, amino acid units. Okay, this the amino acids is formed has two main parts: a carboxylic group, which is acidic group, and amine group, which is a basic group. So this makes the enzyme or the protein is very, very, very sensitive to two main things: the temperature and the pH. The temperature, where the temperature affects the enzyme, and the pH value, which is the concentration of hydrogen ions inside the solution. Okay, they also make the protein, the enzymes are so specific. Why? Because each enzyme accelerates only one type of reaction. Okay, they do not affect the products of the reactants. Why they do not affect the products? As they work as a catalyst, only increasing the rate of the reaction until it reaches its equilibrium. Okay, some enzymes have a reversible effect. What does this mean? This means that any enzyme, as here, the enzyme can return again to make the same job, reverse action. Here, do you see here the arrows? This means that they can return back, combining the products to form another things, okay, or to return it to its original state. Okay, this is the irreversible reaction of the enzyme. The activity of the enzyme is affected by the temperature and the pH. Some enzymes are secreted by the cell inactive state, in an inactive state. So they need certain substance to activate them. Okay, example, pepsin enzyme, which is secreted inside the stomach. They are secreted in inactive form called pepsinogen, and by the help of the HCl, which is present inside the stomach, it will be converted to the active pepsin. Why this happens? So that this enzyme catalyze the hydrolysis of the protein. And we break down protein. And our stomach is formed of muscles. The muscles is formed of protein. Okay, so they are secreted in inactive form so that they cannot hydrolyze the stomach wall. Okay, so let's move to the digestive system in the human beings. The digestive system in the human consists of two main parts. First, first, we know that after we took our nutrition, we knew that the enzymes help the reaction to occur. We have to know about how this food can reach the smallest parts in our body. How can I get benefit from this food? I will get benefit from this food by system. This system is called digestive system. Okay, the digestive system, it is the system which digests food. What's the meaning of digest? Digest, to break down large molecule into smaller ones. This system is formed of two, or consists of two main parts: accessory glands and the alimentary canal, where this accessory glands consist of salivary glands, liver, and pancreas. The salivary glands, which is present inside our mouth, um, under our tongue, and the liver, and the liver, and the pancreas, and the pancreas. Okay, this is the pancreas. Okay, this is the accessory glands, or the glands that secretes enzymes that help in the digestion. Okay, and the second part is the alimentary canal of the digestive canal, which is consist mainly of mouth, pharynx, esophagus, small intestine, and large intestine. The digestive canal starts with the mouth and ends with the anus, which is the ending of the large intestine. Okay, okay. Let's start the digestion. inside the mouth, which is the first part in the digestive system, there are some uh, there are teeth inside our mouth and molars and tongue. Okay, many kinds of teeth are present inside our mouth. Incisors, the first kind of the teeth is incisors. Incisors get the two in the upper jaw two and in the lower jaw two. These incisors are for cutting the food, okay, and the canines for tearing the food, and the premolars and molars for crushing and grinding food. Okay, so the digestion starts inside our mouth, where the mouth we can cut, tear, and grind the food. After that, the tongue. It manipulates the food and serves as an organ for taste. Manipulates the food, mix the food with the saliva. The saliva which is secreted from the salivary glands. What are the salivary glands? They are three pairs of salivary glands uh, under our tongue. They secrete saliva into the buccal cavity, the mouth cavity, through canals. Saliva is formed of mucus and amylase enzyme. This enzyme, which is called a betaline enzyme, this enzyme catalyzes the hydrolysis of starch. أنا قلت لكم from this point we have to يعني remember the word, the sentence catalyze the hydrolysis of. Any enzyme catalyze the hydrolysis of. What's the meaning of catalyze? Help the hydrolysis, the breaking down with the presence of water, of dots and dots. Okay. The amylase enzyme, which is present inside our mouth, is called betaline enzyme. Our mouth, what do you think? Our mouth is alkaline medium or acidic medium? What do you think? Of course, alkaline medium. Okay, the amylase enzyme, which is betaline enzyme, works in an alkaline medium inside our mouth. It hydrolyzes the starch into maltose. The starch is a polysaccharide and the maltose is a disaccharide. Okay, by the help of the amylase enzyme. Okay, so this is our mouth. These are the canines and the incisors. Sorry, are the incisors and those are the canines. Okay, and the premolars and the molars. The premolars and the molars, and with the help of the tongue and the salivary glands, which is under the tongue's three pairs of salivary glands, secretes the saliva, which contains betaline enzyme, help for the digestion of starch and converts starch into maltose disaccharide. This means that the starch خلاص كده is digested completely? No, it is only hydrolyzed from a polysaccharide to only a disaccharide. Still there is a monosaccharide. Okay. Okay. The second passage of for the food is from the mouth to the esophagus, or to the pharynx, first to the pharynx, then to the esophagus. Inside this pharynx is only a passage of the food from the mouth to the esophagus. Okay, okay. Inside the esophagus, which is another tube, connects between the pharynx to the stomach. This inside this esophagus, action of peristalsis action takes place. It is a series of rhythmically muscular contraction and relaxation that extends along the alimentary canal. Okay, this is responsible for the pushing of food through different parts of alimentary canal and is responsible for churning the food and mixing it with the digestive juice. This is how the peristalsis takes place. The food enters, this is the pharynx, and this is the esophagus. Okay, inside it, is contraction takes place and relaxation, contraction and relaxation. The bolus here is the food. After the food enters our mouth, it is called bolus. Okay, the bolus, it will move by a peristalsis until it reaches the stomach, until it reaches the stomach. Okay, what happens by inside our stomach? The digestion in the stomach, which is the gastric digestion, takes place inside the stomach, where the stomach is a sac, muscular sac, okay, which is joined with the esophagus from the upper part, and from its lower part, it joins with the intestine, small intestine. Okay, where the food enters from the esophagus to the stomach by help of sphincter muscle. Sphincter muscle that opens and closes كده. Okay, and يعني narrow muscle here, a narrow joint. This sphincter muscle where is called the sphincter muscle, which joins the esophagus with the stomach, and another sphincter muscle which joins the stomach with the intestine, small intestine. Okay, what happens back inside our stomach? Inside our stomach, back the food enters, where inside our stomach it is acidic medium, where there is gastric juice inside our stomach. The gastric juice is formed of HCl and formed of an enzyme which is secreted in inactive form, which is called pepsinogen. Okay, and the HCl convert this pepsinogen into pepsin. Okay, it is the gastric juice, colorless acidic liquid formed of 90% water and HCl and a pepsin enzyme. The proteins are the only food substances that are affected by the gastric juice. Okay, they can hydrolyzed by the gastric juice. Okay, why does the gastric juice do not affect the epithelial cells of the stomach? This is due to two main things. Number one is the presence of mucus secretion, which protects the cells against the effect of digestive enzymes. Number two is the pepsinogen the pepsin is secreted in inactive form called pepsinogen, which will be activated by the help of HCl. Okay, let's move to the other part, where the function of the HCl, number one, creates acidic medium. Number two, it activates the pepsinogen. Number three, it kills the harmful bacteria that enter with the food. Where we remember that the amylase of the betaine enzyme inside our mouth, which hydrolyzes the starch, works in an alkaline medium. So inside the stomach, I have to make the medium acidic for the pepsinogen to work, okay? For the pepsinogen to be converted into the pepsin and work. So this is the functions of the HCl. Where the action of pepsin enzyme is the hydrolyzing of protein, hydrolyzed as we said before, is plus water. In the presence of protein plus water, sorry, هنا HCl, here HCl, and the other one is, sorry, and here is the pepsin. Here is the polypeptide. البروتين will be formed of polypeptide chain. بس كده اتفك. Okay, is not complete digestion. Okay, so inside the stomach there is no complete digestion, incomplete digestion. Okay, so Again, this is the esophagus, the cardiac sphincter muscle, and inside it, the stomach, where this muscles and the secretion of mucus layer inside the muscle, inside the stomach, and the gastric juice, and the other pyloric sphincter muscle, which is connects the stomach with the intestine. Okay, let's move back to the small intestine. Inside the small intestines, they are the digestion completed. Okay, so stomach incomplete digestion, where the digestion is completed inside the small intestine. The small intestine is formed of two main parts, the duodenum and the ileum. The duodenum is the upper part, and the ileum is the lower part. Its length about 8 meters and its diameter 3.5, very large in length, okay? At its beginning, diameter is smaller, then it will at the end, it will get smaller and smaller, okay? Its coils and loops, they are connected together by a mesentery layer. Its juices, three juices, help to digest the food in the small intestine. What are those juices? They are bile juice, pancreatic juice, and intestinal juice. The bile juice, which is secreted by the liver, while the food passes through the duodenum. What is the function of the bile juice? The bile juice is secreted by the liver. It emulsifies the food. It works only on the fats. Emulsify يعني بتذوب the fats. Okay, dividing large masses of fats into smaller ones. Okay, why? To facilitate the enzymatic effect of the fats, which are insoluble in water. Where, very important note, the bile juice is the only juice that do not contain enzyme. It only works as the enzyme, like it is not an enzyme. Okay. The second juice is the pancreatic juice. The pancreatic juice is secreted by the pancreas, where it has sodium bicarbonate, pancreatic amylase, and trypsinogen. The sodium bicarbonate, it neutralizes the HCl. As we remember that the stomach was acidic medium, with here بقى I need تاني to return to the alkaline medium. Sodium bicarbonate here, it will neutralize the HCl and return the medium into alkaline medium. Where the pancreatic amylase, similar to the amylase of the mouse amylase, but it catalyzes the hydrolysis of starch and glycogen into maltose. هنا بقى starch and glycogen موضوع كبر أكتر by the help of pancreatic amylase in an alkaline medium. Still maltose, still disaccharides. And number three, which is present inside the pancreatic juice, trypsinogen, which is inactive, uh, again, inactive like the pepsin. Trypsinogen, again, is inactive, and by the help of another enzyme called intrakinase, it will be activated and converted into trypsin. Again, the trypsin part 2 is concerned for the hydrolysis of the protein, where the protein are for برضو again will be hydrolyzed into polypeptide chain. The trypsin is very strong than the pepsin. Okay, okay, let's move to the other part. Uh, the lipase. The lipase is the برضو another pancreatic juice. Lipase, it catalyzes the hydrolysis of the emulsified fats into fatty acid and glycerol. Here is the first step in the complete digestion, where the emulsified fats will be converted by the help of lipase into fatty acid and glycerol. خلاص, this polymer will be a monomer, fatty acid and glycerol, and here is completed the digestion of the fats by the help of lipase. All of that happens inside the intestines, the small intestine, the upper or the lower part, of course, the upper part, which is called duodenum. Number three, the intestinal juice. Intestinal juice also part secreted inside the intestine, the small intestine, inside the duodenum. It is secreted by certain cells in the wall of the small intestine. It contains several enzymes. بقى الintestinal juice بقى جايه تخلص كل الفود. Intestinal juice will complete all the digestion. Okay, all the food, the remaining of food will be digested, will be break down into smaller units. The first one is the peptidase. Peptidase will have the polypeptide chain اللي هي جايه from the protein polypeptide chain. Then peptidase will form amino acids. Okay. Again, disaccharides enzyme any بقى disaccharide maltose with the help of maltase is يعني enzyme. Maltase, glucose and glucose. Sucrose by the help of sucrase, glucose and fructose. Lactose by the help of lactase, it will be converted or hydrolyzed into glucose and galactose. Okay, where برضو there is an intrakinase enzyme which is present inside the intestinal juice. This intrakinase enzyme, it is not a digestive enzyme, يعني it doesn't catalyze the hydrolysis of any of the food, but it acts as a coenzyme that activates the trypsinogen to be trypsin. Okay, where the trypsin works on the protein and form polypeptide chain again. Okay, okay. And then let's talk about the absorption. Absorption. After the food reaches the small intestine in the upper part of the small intestine, which is the duodenum, it will then go to the lower part of the intestine, which is called the ileum, where here absorption takes place. كل ده خلاص the food is digested. Then what will happen after digestion? It must be absorbed to the blood, okay, so that it can pass to all parts of the body. Where what is the meaning of absorption? It is the transfer of the digested food substance to the blood and lymph through epithelial lining, mucosa, epithelial lining of the small intestine. It takes place by the inner epithelial lining of the ileum, where the inner epithelial lining of the ileum is folded to form villi. The villi increases the absorbing surface area of the inner lining wall of the ileum, rating to reach about 10 meter cube. That's to say about 5 times as much as the surface area of the human body. Oh, okay. Why this for increasing the surface area to increase the absorption. Okay. The structure of the villi is very important عشان to know how this villi works. The villi has epithelium. This is the small intestine, the ileum part. When we take a look inside it, we will notice that there is projections coming out from it. Okay, this is what it looks as in real. Okay. Okay, in the villi consists of epithelial layer, and inside there is this epithelial layer. Inside there is a lymphatic vessel, like lacteal vessel is surrounded, lymphatic or lacteal vessel surrounded by blood capillaries, network of venules and arterial blood capillaries. They are very tiny microscopic projection. Those villi are very tiny projection. They are called macrovilli. كمان there is a macrovilli inside them. Why this macrovilli are present? To برضو increase the surface area of absorption. The product of the digestion is then transferred through the villi to the membrane by diffusion and active transport. Okay, they pass in 2 main routes, blood route and lymph route. Okay, this is the blood capillaries inside the villi. Okay, this is the villi. We took this part and we enlarge it under the microscope. We here there's projections, okay, and this part is the microvilli. This how it it is present in real. Okay, this is the villi. So and then this villi will take the absorb the food and pass it to this network to go to the whole body through blood route and lymph route. blood route and lymph route اللي هو اليالو route اللي هي lacteal vessels. Okay, so the blood route, it starts with the blood capillaries inside the villi, They absorbed substance, the water, minerals, monosaccharides, amino acids, vitamins which dissolve in water will go to the blood route. These substances are carried to the hepatic portal vein of the liver and then to hepatic vein, then to inferior part of the vena cava of the right atrium of the heart. Okay? Ta, 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 ta. Okay? This is how the blood moves. Okay? And the other part is the lymphatic route, where it starts with the lacteal vessel inside the villi. فاكرين اليellow part, lacteal vessel اللي هي اليellow part دي, lacteal vessel inside the villi, where this lacteal vessel will absorb only fatty acids, glycerol, vitamins which dissolve in it, اللي هم adic vitamin A, D, E, K, adic vitamin, some fats. The fats are resulted from the recombination of some fatty acids with glycerol. Some fats كمان بترجع تاني again لأن في fatty acids and glycerol will return back to be a fat again. Okay, this is how it looks. This is the arteries and the veins. The artery carry the oxygenated blood عشان كده its color is red, where the veins carry deoxygenated blood. So we always color it as blue. Okay, this is the lymphatic capillary, and these are the blood capillaries. Okay, how the blood flows in the human circulatory system. Some emulsified fats which are not digested by the lipase enzyme are absorbed directly by engulfing by the epithelial cells. Okay, these substances are carried to the lymphatic system, which carries them slowly to the superior vena cava, okay, upper part, then to the right atrium of the heart. Okay, this is how it looks. The oxygenated blood comes from the heart, goes to all body system, then exchange through the blood capillaries. They take the oxygen and remove the carbon dioxide, then return through the veins to the heart. Okay, this is the lower part of the body and this is the upper part of the body. Okay. Okay, thank you.